This is the presentation on basic R&D to commercialization and how to. It's a presentation that I gave for the university, Quest International University, as a campus-wide presentation to make the researchers understand by using an example. The example is me and how I progress or develop three basic research projects with the first two unable to lead towards commercialization, but the last attempt managed to achieve commercialization status. So it is giving my experiences on these three attempts until successful leading towards commercialization. So this slide gives an idea that not really everyone in a university research institution able to take their R&D to that commercialization status. In fact, if I can make a comparison in my previous engagement in a government university, if there is around 500 active researchers, it can be said around 5 to 10 only of the active researchers have gone to commercialization status. And if they do, they usually follow or apply this technology readiness level, TRL, and this is TRL that is been adopted from what NASA has done in 1970 for their ability to progress any project from A until Z successfully, efficiently, without major problems. Okay, but this NASA's TRL was also adopted by, as you can see, by MOSTI in order to get researchers to plan from TRL 1, which is basic research, up to TRL 9, which is commercialization. So the takeaway message is not everybody able to uh, progress towards TRL 9 from basic research. There's actually plenty of resources. If you are a researcher that is more specifically bio-related researcher, you can work on a lot of resources related to plants, mammals, and until microbes. In terms of checking the biodiversity, the diversification of each of these categories, you can say there are plenty. At the world level, you can see vascular plant, which includes medicinal plant, herbs, plenty. And then our country, Malaysia, also have plenty of diversification when it comes to tropical plants, can be also medicinal plant. If you're looking into microbes, it's always more surplus of microbes available for, doesn't matter, microbiologists or non-microbiologists looking into working with microbes. The challenge is not having a lot of resources. The issue is getting hold of that material, and it's always been the challenge. For instance, in my research working with snake venom, there are around 2,000 species, and one-fifth are venomous snake venoms. So I have to get hold of the snake, but I am not trained, and also I am not brave enough. So I have a snake handler I have employed when as and when required. He will get the snakes, and also well, he goes into the forest to get the snake, or whenever possible from people who have stumbled upon these snakes. He will get them and then will call me up. I will go to meet him, and then we will milk the venom at the spot. So the challenge is so much of resources to work on, but getting a hold of that material and also at a quantity useful for the R&D and later commercialization, because commercialization definitely require plenty of that material. To take an example of a researcher who best I can use than myself. So this is my achievement being attached to a public university in Malaysia for past 10 years, that is up to 2024. And in that 10 years, I have got this many publication, 50, and also citations of slightly more than 1,000. For those who are new, will realize that 1,000 in that particular public university is actually at the upper level, more than halfway point, and at the 75% of the achievement in terms of citation for all the researchers. So I know I was in a good status when it comes to citation. Okay. And patent-wise, three applied, two completed. Actually, before I left, the third one also is in a good status of getting approved. And of all the three patents, one actually was commercialized. That is, I wasn't the PI. My friend was the PI, and I got a bit of money from that commercialization effort. And the number of products that I have commercialized are to be two, and both are related to Tongkat Ali, medicinal plant. And then in that 10 years, I have achieved 400,000 total sum of money from various grants. Get directly into the first attempt, which is a project related to milk coagulation. I got the idea of this milk coagulation by looking at a video from, I think it's Chiang Mai in Thailand, where they were milking venom from a snake and then immediately putting the fresh venom into fresh blood. I'm not sure where is the source of the blood. And upon some time, very fast, the blood got coagulated, became jelly-like. So I know that is possible. I was thinking, what if I do the same by taking a venom that I have in my storage and putting into fresh milk? What will happen? That's where I did it. Uh, and then I saw the following day when I came back and checked it, the milk already coagulated. So that gave me an impression I was in the right direction. It is possible to use milk venom to coagulate milk. So why I want to do coagulation of milk? Basically, it was found that rennet, or also known chemosine, this rennet is the commercial used enzyme to coagulate milk, either at cottage industry or in a upscale, bigger industry level. Okay, for bigger upscale industries, they will use rennet that has been genetically modified, genetically obtained by using GMO or LMO, genetically modified organism or living modified organism. But the issue is the cheese or yogurt obtained have to be labeled GMO, LMO product. This keeps away certain health conscious people who think it is not safe to consume these cheese. and therefore sales may not be as easily obtained as compared to using rennet from the stomach of young animals. Usually they will use cow, and therefore the young animal for cow is calf, and then goat, the young animal for goat is kid. Also from pig. Yeah, you heard me right. It's from pig as well. So when using all these young animals, one thing getting from the stomach, very little can be obtained. Therefore, better go for GMO, LMO produce rennet. And when using from the young animal, ethical issue comes about. You are sacrificing a young animal before it can become proper adult, but have to use young animal because Rene is only found in young animals, that is not or unweaned animals. That means not taken away from their mother. They still rely on mother's milk. And since I said also coming from pigs, there is a religious implication there. Okay, so anyway, having all this into consideration, I still went forward with trying to work with snake venom and coming out with new resources or new source of uh, coagulant enzyme. Slide, you can have an understanding what are the sources for rennet. It could be from animal such as rabbit. Porcine is actually pig, and cow also should be here. Can also from plants, but more popularly animal, and also from microbes rather than plant. Okay, so these are popular types of microbes. They can obtain the rennet or chemosid. When it comes to porcine or pig, uh, there is actually in Italy, traditionally, many centuries, these cheese been produced. The issue is if Muslims are not aware, they might end up purchasing and then knowing it is actually using only the coagulant, not the milk. The milk does not come from the pig, but actually comes from cow milk that has been put with coagulant taken from the stomach lining of pigs. So that's how little they need to put. And I'm not sure whether by using DNA or molecular detection method can actually detect so little of enzyme put in a vast amount of cow's milk. So have to be careful there. May not be detectable in any scientific method use. And then a bit on how milk coagulation takes place. If you have milk, within inside it, there is this kappa casein. This kappa casein is a micelle, a ball which has a protrusion, like hairy protrusion, and then by using this rennet or any other enzyme, protease that can cut off all that protrusion, the hairy part, it will be then smooth as shown here. When it becomes smooth, it cluster together or like a glue comes together, and that is called curd, while all the other liquid portion having no uh, not becoming precipitant, not curdling, are known as whey, the liquid part. So once coagulated, milk will become curd, semi-solid, and also liquid part, which is whey. All the 11 snake venoms under my possession that has been freeze dried after taking from the snakes over the years. And then we tested by putting each one into fresh milk and looking at the time taken to coagulate. Okay, later I'll show you a video how that is done. We can visibly see milk turning into curd and whey. Okay, and the one that has shown to be quite promising with the fastest time of 2.83 minute is that of Callouslasma rhodostoma, or also known as Malayan pit viper. So this is really uh, found in Malaysia, and it was taxonomically named as Malayan pit viper, okay, or Callouslasma rhodostoma, a viper, and is also commonly known by Malays as Ula Kapa Bodo. If I have time, I will explain why is it called Ula Kapa Bodo. Not to say bad thing about the snake. It's actually something related to the snake bite and not going away, staying there close to the victim, and that's why it's called Ula Kapa Bodo. To make a long story short, it took one whole master's students 2 years to actually do this project. Once we have found the best venom to coagulate, very short time, 2.83 minutes. We then took that venom and started to isolate into fractions by using two stepwise chromatography techniques. One is ion exchange chromatography, and then we get a lot of fractions, and then we also entered into size exclusion chromatography. This is referred as bioassay guided fractionation. The bioassay is the milk coagulation capabilities. Every fraction is tested for milk coagulation capabilities, and then based on that technique, we managed to find only one fraction, and that is this one. So the Malayan pit viper venom has so many proteins in it, and this lane shows a lot of proteins. Some could be very toxic, but the one that able to coagulate milk when we tested the fractions, only this one, okay, which has we put also standard. The molecular weight is between 30 to 20 kilodalton, to be exact, 26 kilodalton size wise. So we have managed to purify, and this is the one giving milk coagulation in this venom. Step is to take the purified protein, send for identification by using LC-MS. This was done by a service provider, and then the results came out to say that it is histamine, shown here, metalloproteinase histamine to be exact, and it is from the this same snake venom of Malayan pit viper. And this is the modeling structure of this histamine. So can be said we already purified to the single protein from the venom for the coagulation of milk. We have managed to purify the protein that is related to coagulation from this snake venom, and also identified it. So it was exciting. However, to think further, to take up this R&D achievement to commercialization, we realized some issues, some challenges. One being the scarcities. How many snakes do we have to, extract the venom in order to get enough sufficient amount to scale up? So that was an issue. Another one, still the stigma of people thinking, the protein coming from snake venom, and therefore whether it is going to be uh, having to be poisonous for them to consume the product, which is the yogurt or cheese made from this enzyme itself. So that's another thing. We can also resort to having genetic engineering of the enzyme, that is found in this snake venom. That will take some more time and expertise that we need to recruit. So, thinking about all this, naturally decided to just not abandon or abort it, keep it in view for future, and look into other projects. Having the first attempt facing challenges after accomplishing the purification of the coagulant from the snake venom, it was left to try the second attempt, another project. But since already established the assay of milk coagulation, having a lot of consumables and also chemical related to the other project, the attempt one, was much easier to diversify to this second project, which is a useful application of milk coagulating from plants. So this is good because if utilize plants, and therefore it is considered to be halal, and also vegetarians wouldn't have any issues as well. What is good also, no need to purify the coagulant or the enzyme related to milk coagulation, especially because medicinal plants are having phytochemicals that can give benefits. The benefits included antimicrobial capabilities, anti-diabetic, and also antioxidant. So with that in mind, having 14 plants been sourced based on phytochemical activity, these three phytochemical activities, that is antimicrobial, anti-diabetic, antioxidant, not directly related to milk coagulation capabilities, because not many plants out there having milk coagulation been done, so not much literature on that. So we go about looking into the phytochemical capabilities instead. The similar manner that is done on the snake venoms. Again, we looked into milk coagulation capabilities, the coagulation time by putting milk. And from the 14 plants, stands out. These three are noni or Morinda citrofolia, and then pineapple, the variety of Sarawak, and also mango, which is only one type of mango, the apple mango. We tried other types of mango, did not give coagulation or milk. So the rest either have not so good coagulation time or not at all. So to go forward, instead of choosing the best one, we decided to combine the three best coagulating time, the three ranked highly. We combine it and we call it supercoagulant, right, with a ratio of 1:1:1:1. So a bit of information related to supercoagulant. The ratio is 1:1:1, and noni, pineapple, mango. Among these three, pineapple already have enzyme in it. It's called bromelain, and bromelain has protease activity. That means can cleave the casein, the micelles of the casein, and cause coagulation. The other two having lack of information ever been uh, investigated for milk coagulation. So we combine three at the ratio of 1:1:1 just to build up our, thinking that it is related to enzyme, not due to lowering of pH. We actually did protein assay, Bradford identification or quantification of protein, and we saw there were protein concentration in this supercoagulant itself. Therefore, encouraging to know that it is could be enzyme related not because of lowering of pH. So in this slide, I will demonstrate to you how when supercoagulant is put into milk, it is able to cut the kappa casein, therefore removing the micelle protrusions, micelle hairy protrusions, leaving behind the kappa casein to become curd. So let me press the button here. This is observe milk after putting supercoagulant at light microscope with 100 times magnification. So as you can see, it is confluent, but as the curd is formed, there will be gaps, and this is the whey, while this is the curd already formed once the the hairy part is cut off, and it will form the curd. So this is a good video demonstrating how curd is formed once the supercoagulant is added to the milk. So here there is in the middle, that's where the light microscope. This is how milk looks like, all confluent, liquid, and then this is how the semi-solid formation of curd after the supercoagulant has been put into and leaving it for a while after mixing, and also the rennet. You can see gaps similar for supercoagulant and milk, and also milk and rennet. This is same as the video on supercoagulant and also milk. This one is put in an Eppendorf tube, milk alone. And then this one, after some time, the supercoagulant and milk. And then we spin it at 1000 RPM for a few minutes. It will form the curd at the bottom. All the sediment is curd, while whey is in the top. Similarly, milk and rennet, the curd is here while the whey is on top. You can see it's clearer than the milk plus supercoagulant because the phytochemicals have color. So it actually enriches the curd with the phytochemical. This one is actually, we took the milk, froze it, and then freeze dry it, and then sent for SEM, scanning electron microscope at high magnification, much higher than light microscope. Similarly, we took the curd, freeze dry, and then sent for supercoagulant plus milk, and also milk plus rennet. You can see the view or the observation looks like very concentrated, congested, while the milk alone having much more gaps and more spaces compared to the milk supercoagulant curd and milk rennet curd. Moving forward, we took the curd that is formed by the supercoagulant plants and then do some tests, biological activities. Some included phytochemical determination in the curd, whether there is any activity relevant constituent using LCQ, TOF, MS, using libraries relevant to medicinal plant. And then antioxidant activity, we use the common one, DPPH and also FRAP. These are the common for antioxidant activity. We also did anti-diabetic determination. You're looking at these two enzymes, also commonly done. But we didn't stop at the in vitro. We also progress at in vivo. But before we did in vivo, we determined whether the curd obtained by supercoagulant has any microbes, unwanted microbes that can cause blood sepsis for the rat if we are going to create uh, diabetic rat. We also look at heavy metal found in the supercoagulant plants, whether there is any heavy metal. And lastly, we also did antimicrobial determination using agar-well diffusion method. Based on the phytochemical determination, we found out that the supercoagulant curd, the curd not supercoagulant, The curd has antioxidant activity, the anti-diabetic activity, but missing was antimicrobial activity for the curd. So it was apparent the although the literature say got antimicrobial activity, antioxidant activity, anti-diabetic, but when we actually tested the curd, antimicrobial activity was actually missing. Moving on, we did biological determination, the antioxidant and also anti-diabetic, and the next slide is antimicrobial. For the antioxidant, you can see the supercoagulant curd and also rennet curd was investigated using DPPH and also FRAP. For DPPH, we had the gallic acid as the standard to compare. And you can see the IC50 value. The lower the IC50 value, the better it is as an antioxidant. And it provided 26.53, 85.35 for this assay as well as DPPH assay. To take DPPH an example, you can see you can't beat gallic acid because IC50 was much lower. Nevertheless, there is IC50 determined for supercoagulant curd compared to rennet curd having no activity at all. So there is no phytochemicals in the rennet such as that to give antioxidant activity. While anti-diabetic determination, we use acarbose. It's a very old medication, so not as effective as the current one. Nevertheless, we purchase it from the pharmacy and test it for its IC50 to go against this alpha glucosidase and also alpha amylase. The supercoagulant curd can't beat acarbose. Nevertheless, it showed some IC50 values for both alpha glucosidase and also alpha amylase. So it can inhibit these two enzymes, which are related to increasing glucose in the blood. But again, rennet curd did not provide any activity. The antimicrobial determination, we use this whole diffusion method where we punch hole. Before that, we pour the bacteria or fungus. So we tested for a number of gram positive, gram negative, and also the fungus and also yeast, selected ones. And based on the outcome, the results, using this method, where we measure the diameter, the gentamicin obviously showed positive diameter inhibition zone. Uh, but supercoagulant curd, the green one, not to be found on any. No gram positive, no gram negative inhibition, and also no fungi strain inhibition. It's supercoagulant or the plant material, we're not talking about the curd. The plant material, nevertheless, has certain inhibition capabilities for a number of gram positive, five of it, as shown here, and also one gram negative, but not on the fungus. So we thought for a while, why not we refortify? Refortify means once we get the supercoagulant curd, the curd it was added with the plant material, the supercoagulant, and then tested for the whole diffusion method. When we did that, we realized the antimicrobial activity is restored. So we start to get the supercoagulant refortified curd having similar activity to its original plants. So what it means is, so yeah, the supercoagulant curd has antioxidant activity, diabetic activity, but if we want to have antimicrobial activity, we have to convert it to curd and add the supercoagulant after that. But we were satisfied with two activities alone, anti-diabetic and antioxidant. While this one, we save it if necessary. Also created our diabetic rats and then tested the anti-diabetic capabilities of the curd. how we did was we got the rats to be injected with this STZ, which will make the insulin ineffective. So it's a diabetic rat ready. And we also fed with the oral gavage of the curd mix or dissolved in a right media, and then gave it on the morning and also at around 5 o'clock every day to the diabetic rats, four diabetic rats, and also had another group for diabetic control, all diabetic rats, monitored. Every day we monitor their glucose, fasting glucose level by using a glucometer. So this is the outcome. As you can see, the blue is on what do you call normal control. No changes, around 7.5, slightly higher than humans, but that's what we got reading from the glucometer. And then it for the diabetic control increase and maintain at 26 or 25 throughout the 20 days of monitoring. While the one that's been given the supercoagulant curd every day, morning and at 5 p.m., quite good. It started to drop over the period of 20 days. It became at the last measurement around 15. Uh, sure, if we progress more than 20 days, it would have dropped even further. But at 20 days itself, we were having issues where the diabetic control rats were losing a lot of weight. So it's best to stop the study at these 20 days. But encouragingly, it showed that the supercoagulant curd has anti-diabetic activity at in vivo level. It was exciting to see that the second project also, in a way, successful at R&D level. So what we did was we bought this homemade yogurt maker that you can obtain from Lazada or Shopee, and then we put milk inside, fresh milk, added the supercoagulant at a ratio and also weight calculated, and left it overnight, and then made it into yogurt drink by adding a little bit of flavoring and then having various types of yogurt. So, why is it not available commercially? To answer to that, again, we experience problems where we cannot go commercial level simply because the taste of these yogurt drinks was bitter. uh, I tried adding certain flavors to remove the bitterness, and then looking at the literature, there is a chemical compound or constituent that is once coagulated by plant coagulant, that constituent will appear, and to remove it is very difficult. Uh, by adding honey, it seems to have reduced in the bitterness. But I have to ask you the question, if you're coming up with an anti-diabetic supplement such as this yogurt drink, not advisable to actually add sugar, right? So honey is sugar. Therefore, uh, looking at the challengers, decided to abandon this project as well, at least for the moment. Move on to the third project, and this one considered third time lucky, and this one is on the Tongkat Ali plants for boosting testosterone, the male hormone. There are a number of Tongkat Ali plants out there. The one that I have looked into is three types. Actually, there are more than three, five. Uh, but at the moment, not sure what are the other two. So, uh, what I can get hold of is these three types. They are the white variant. The root is actually white or yellow, so they are known as Tongkat Ali Putih, white color. And a lot has been studied on this type of Tongkat Ali. And then you have the black one, simply known as Tongkat Ali Hitam. And then you have the red one. The root is actually red in color. So it's called Tongkat Ali Merah. All three having genus and species which are different from one to the other. One is Eurycoma longifolia, the other one Polyalthia bullata, and the last one Stemma tuberosa. And looking at the white variant, which has been studied more than 50 years, it has many products out there. It's not only confined to Malaysia and surrounding region, but it's already gone international. If you just put in Amazon.com, there are many products available for Tongkat Ali. The slide shows you what are the traditional usage, traditional potential for these three plants, and also what has been studied so far on the plants. The white variant, a lot of traditional uses. Most popular is the aphrodisiac. That means useful for libido, useful for sexual performance, useful for sexual dysfunction. Okay, that's aphrodisiac to you. All three has traditional aphrodisiac claims. And then the scientific paper, a lot on the white variant up to clinical trial, from basic research up to clinical trial has been done on the white variant. Also already isolated the compound relevant to aphrodisiac capabilities, while it has been already clearly investigated, able to boost testosterone, ATP, many other things. But the two are lacking in studies or very little studies, and even what is being sold only in the form of slice, not yet capsulated into what you call traditional preparations. So that gives us room to investigate, especially for the two other Tongkat Ali variant. Did for our R&D, we went and get all the three types of Tongkat Ali from reputable sources, which is indigenous people, pulverized, extracted, freeze-dried, and then the powder become capsule. And then we tested the capsule for all these quality, uh, which I will show you the results in a while. And the capsules that we made, we tested by seeing whether it can elevate testosterone in a chosen animal, which is fowl or rooster. More details on what we did. We collected the plant, the roots from indigenous people. We also authenticated by putting voucher specimen in University Malaya, their botany voucher specimen unit. This is the code given for the voucher specimen. So here shown more details on how we went about processing the root materials, three plants, roots. We cut them, chop them into small cylindrical shapes, and then using this medicinal plant or Tongkat Ali medicinal cutter, they call it. We slice it into chips, and then we blend the chips, and then we sieve it into fine powder. I was also given some money to buy all this equipment, instrument. Next, after we get the fine powder, we actually extracted using this device, where at the bottom, round bottom flask, we put the water and the fine powder of the Tongkat Alis. Each one reflux for 5 hours with water, and then we sieve and obtain the liquid alone, throwing away the debris. extract liquid is freeze-dried. We then tested into the pH, the moisture content, the heavy metal, whether got steroids or not using GCMS, and also microbes in the freeze-dried extract material. So this is the results of the test, all three. This is the white variant, the black variant, and the red variant. You can see the freeze-dried extract material. The pH is around 6, this also 6, this also around 5. So it's not a concern to actually adjust the pH to 7 because we also check ginseng is around 5. So we had in our experience last time one more plant, they call it Terminalia catapa. When we extracted with the same way, the leaf, it was having a pH of 2. So that one is a no-no to go forward. But this is around 5 to almost close to the neutral pH. So we didn't do any adjustment. Moisture-wise, we also check with ginseng and other medicinal plants. It is on a low level, so it's okay to progress. It is not likely to get spoiled because the moisture content is very low. Heavy metal, since we got it from the wild, indigenous people managed to get it from the jungle, away from any industry area. Therefore, there is no heavy metals were detected, so it's safe. Steroids, also no steroids that we found. In fact, we also tested for testosterone, and there was no testosterone found in any of these three plants. So whatever that is causing it to elevate testosterone is coming from some kind of phytochemical or other constituent found in these plants. The only negative was microbial detection. There were microbes found, especially for the total aerobic microbial count and also total yeast microbial count. There was numbers, values given, causing us to worry if we put it into the rooster or the fowl, that will cause some unwanted effects to the fowl or the rooster. So what we did was we sent it for gamma radiation and therefore done the decontamination. Once again, we tested, there were no detection of any of these microbes. It came to the in vivo testing by choosing fowls. Choosing the fowls also need to choose a certain weeks old. It has to be between 26 to 28 weeks old, because if it gone around 30 to 35 weeks, it's already reaching the fertility peak. So better to select a bit younger. And then the material, the tongkat ali extraction, we made it into capsule by using a capsule maker that we purchased from Lazada or Shopee. And the dosage method, it was a bit challenging, whereby we read what was done by others. It's supposed to put the capsule at the beak, and then cause the rooster to swallow it. So that was not easy. However, with the help of the farm owner, it was achieved. This is how we did it. Yeah, let me put the video on. Hopefully, the video plays in the recording. So, what we did was we opened the beak, open the beak, and then put the capsule into the mouth, and then using a distilled water bottle, squirt the water, and then it will start to swallow. So we monitored few parameters during this time. How we gave the dosage was one capsule in the morning and one more capsule in the evening. And we did this for 30 days. for all the Tongkat Ali separately with four fowls for each. Also got control. But after completing for 30 days, we realized the black variant of Tongkat Ali didn't give any positive result. But we remembered the indigenous people say this particular Tongkat Ali takes longer time to show its effectiveness. So with that in mind, we actually did another experiment. After 30 days, we also did for 60 days for that particular Tongkat Ali hitam only, the black variant. But the results of the white and also red variant is as shown here for 30 days. No bad things happen when it comes to the weight of the fowl, because if the weight drop drastically below compared to the control, the untreated one, then we have to worry. But all doesn't matter for 30 days or 60 days, all Tongkat Ali showed progression or increase in weight. So that's good. For the sexual mating behavior, once we introduce a hen, female chicken, we observe certain parameters whether it is going to lead to intercourse. Of course, we didn't look into intercourse or ejaculation. We only look at these parameters leading towards that direction. Whether it happens or not, uh, uh, we didn't go all the way. So there were a few parameters: wing flapping, body shakes, crows, and pecking were the one we calculate or tabulate. And then we sacrificed the animal. Immediately we took the blood, 5 ml, and sent to diagnostic lab, determine the level of testosterone. We also took the liver and testes, put in formalin, and sent to our partner to look into pathological damage, if any, or also activities in the testes. After mating the treated fowl or rooster with the hen, female chicken, this is the outcome based on observing the number of parameter, the sexual behavior parameter, that is wind flapping, body shakes, crows, and pecking. We saw that the control had these numbers, while a slight increase, not significant for the white variant, the red one, and also the black variant. This one after 60 days, while these two after 30 days. So, uh, obviously, this result is not significant, but shows slight elevation. Rely on only one experiment or one test alone. We also took the blood plasma, and started to send to the diagnostic lab to see whether the testosterone has elevated or not. So you can see the control around 4 uh, nanomole per liter concentration, while the white variant increased to 7.7, the red variant 6.25. You may say the black variant has a high testosterone concentration, but remember at 30 days it did not have elevation compared to control. Only after 60 days it is increased to 9.72, and it's impossible to rank it or to claim that the black variant is the best, because this value is for 60 days, while the white and black red variant only done for 30 days. Also determine several biochemical parameters other than testosterone. We didn't have any standard to compare. Unlike humans, we can compare the ALT normal range, AST normal range, but for this we cannot. We just have control for controls for a normal untreated animal, while for the Tonga Ali's, we observe based on the control values, only one parameter, which is the ALT, alanine transaminase enzyme level, was alarmingly high, three times more than the normal control values. And you can see by this graph as well, three times higher compared to the red and also black Tongkat Ali. That was worrying, but as we looked at histology results, we came to a conclusion, and I will explain in a while. Here we also send the testes, liver after sacrificing the fowls, and histology was done on the tissue and graded by the pathologist. Based on the pathology grading and assessment, accordingly, this is the testicular tissue for normal. This one for the white variant Tongkat Ali, this one, the red one, while this one black. So accordingly, the pathologists seem to have opinion there is increase in testicular activities, Leydig cell and also Sertoli cells having more activities compared to the control, while that of the liver, you have to recall from the previous slide, there was an elevation of ALT. But according to the pathologies on the control liver tissue, the Tongkat Ali white variant and the red variant and the black variant, the liver tissues, uh, nothing of uh, extreme uh, effect was seen, or untoward effect was seen on the liver of the white variant treated tissues. If any, only mild outcome. So this gave some idea that we are actually having too much of the dose, high dose for the white variant. So instead of abandoning the whole idea of using Tongkat Ali Putih, because Tongkat Ali Putih has been sold widely, so what we did was we lower the concentration and then decided to develop the product next. So with the whole basic research out of the way for this project on Tongkat Ali, and it was encouraging. And what's next is basically to go up in the technology readiness level from that of basic research and trying to demonstrate whether the product that is being developed, prototype from prototype to pilot scale and then to actually product ready to be marketed. Obviously, we need to get an idea whether it's can be sold. So uh, why not create the prototype first, then use it to demonstrate it can be sold or not. So that was what we did, and we will also need to decide which of the three Tongkat Ali need to go to that prototype level. So I chose actually what has already been done. There are many products on the white variant, so I chose the white variant to mimic it first, to use the popularity of the white variant rather than going for the not so popular other types of tongkali. So the white variant was chosen to become a prototype capsule herbal product. So this is our prototype. It is done in with label, but all made by myself, and then got the printers in Brickfields to print it, and the wording, everything done by me without consulting any ministry on what should be the right labeling. Nevertheless, it was completed, and then using a capsule maker, we made the capsule, some 60 capsule per prototype, meant to be taken twice a day, in the morning and also at night. Uh, and the amount was a bit reduced by calculation, so that nothing alarming will happen to ALT level. So that was lesson learned and applied here. And this prototype, still under R&D achievement, we achieve a lot of things. As been put here, several internal grants were obtained for this prototype or its content. We won medals in exhibition within UMP or within the public university itself. Got ISI Scopus Journal, and also articles, in-house articles, number of postgraduate as well as undergraduate achieve. But all this is still at the low level of the technology readiness. So we went to next level. So to have a very good idea whether the prototype even capable to work, uh, capable to attract attention among customers. So this is what we did. We actually got 20 participants. My student found these 20 participants, and they turned out to be all male. Of course, this product was intended for male consumption, male buying it, because testosterone is more relevant for males after the age of 40. Okay, so happen demographic wise, all of them are Muslims, and these are the age bracket, all five individuals for each of the age that we managed to obtain, and some around five of them actually have tried some sort of Tongkat Ali product previously. Upon using questionnaire and also consent form to get consent in doing this survey, nothing invasive. Everything was using questionnaire based study, where we gave the prototype to each one of them, one bottle having 60 capsules. Before we start the experiment, we actually asked these questions, six questions. We can't ask questions directly related to how well they're doing in their sexual life, or personal relationship related to at the in the bedroom. So we made it a bit masking, and we asked certain other questions first, because we also know Tongkat Ali able to elevate ATP energy. So we asked those questions earlier, and we also know that lowering of testosterone will disturb mood. So uh, uh, we also asked those questions. So six question was formulated, and before we start the experiment, we asked this question, and then after that, we ensured that they took the doses accordingly, continuously over the period of 30 days. So this was the output results after 30 days, and also before 30 days, pre-trial and post-trial. In the pre-trial, if you look at the score for the six question, mostly were yellow, gray, and orange. Dissatisfied, and then neutral, and also satisfied. They don't want to go more than that. But after 30 days, we asked back this question: more blue, which is very satisfied, more yellow than gray was actually achieved. Therefore, a bit promising that the product actually works for them, and they seem to have a better energy level, better, a bit capable to do their work. Mood is okay, and also their sexual life also can be said okay based on the output of result we obtain. This is the benefit that you can say about testosterone. What we understand literature-wise, testosterone is an important hormone, not only for males but also females. Males, they actually have higher level of testosterone in their blood, and women have one-fifth of the amount found in males. Okay, they also have, but very low level. And another understanding is over aging, adulthood, testosterone get boosted, get optimal, and then after 40, it actually 40 to 45, it start to drop 1 to 1.5% over the age, up to 70, 80, it drops. And there is a normal range, and I can show you here serum testosterone tested by a reputable diagnostic lab. They put there the normal range at 6.6 to 25.7, and this individual that we managed to get to know what is their testosterone level. This individual is age 54 years old, and he is actually having normal level of 11.5. So within the normal range. If the person is normal range, no problem. They will look, happy, confident. Muscles are still all functioning okay. But if it is lower than this range, you start to have bone problem, increase in fat, even the side effects is their sexual performance and even dysfunction. And when there's low testosterone, have no interest for sex altogether. So all these, so it's not about testosterone is not about sexual performance, sexual dysfunction alone. You can see it's a wholesome effect for other things as well. Okay, so overall, this person looks sad, while this one much more happier and healthier because of normal testosterone level in their blood. Still in the technology readiness of 2, 3, or 4. We still looking into how good is the prototype. So what we did was, after doing the survey with the 20 individuals, next we actually made into a number of product, the prototype, and start selling. Actually start selling. So we have price at 30 ringgit per bottle. Later you'll see it was double the price of this when we went for production, but now we sell at 30 ringgit and see whether people start buying. We put in Muda, we put in several booths exhibition. We put a booth and have certain posters and welcome people, and then try to convince them to purchase and try. And also we give instruction how to utilize it. The important thing is the instruction. Then only they will see the elevation of testosterone. We kept telling the customers, you can't expect elevation of testosterone to take place in 1 or 2 days. So be patient and keep on using it for at least 20 to 30 days. And based on that outcome, certain customers were actually satisfied, and they end up buying again. So they became a returning customer. So that was positive, and therefore we progress to higher technology readiness level. The next TRL level where we actually register officially and to obtain this MAL number. This MAL number at the far right at the end is T. T is referring to traditional medicine, and in order to sell without any uh, legality issues in Malaysia, you have to register the traditional herbal product to Ministry of Health, and more specifically in the Ministry of Health, there is something known as NPRA, National Pharmaceutical Regulatory Agency. So we then contacted them and start the process of registration for MAL number, step-by-step process for registration that I can recall, and I already put it down. And it took a long time actually. To get the NPRA to register, you need to set up a company. So you have to go to SSM. SSM stands for Suruhanjaya Syarikat Malaysia. And then you have a company form. This is my company that I form. Then you also need a contract manufacturer. So we appointed this Dongfeng Manufacturing because they have the GMP production that we don't have. So you have to have that. And then we started the process with the NPRA together with the help of Dongfeng. We did the purchase of the USB security token in order to use the NPRA website, design the label, which you will see in the next slide. And then accordingly, NPRA through Dongfeng, they want us to test whether got any heavy metal and also microbial load in our raw material before they can approve. And then we answered many more queries about our plant that we are using. And lastly, after more than 6 months, never less than 6 months, I was thinking it will happen in 1 month, but actually it took more than 6 months to 1 year to get the MAL number approved. This is the label that we finally designed. Actually, had to pay someone, freelancer, to come up with this label. You can see that the this is the company form with SSM. This is the contract manufacturer in Kajang, Dongfeng. And then you can see all the wording does not have any scientific bearing or claims which are very health related. Is all traditional purposes. And then, of course, you also have our newly approved MAL number included in the label. And later, when we actually send to Dongfeng the raw material, they will put it into capsule, into a bottle, and put this label, and they will put the official hologram meant for this product. Is the first batch manufactured. Unfortunately, Dongfeng is very required only minimum of 1,000 bottles to be made, and we followed that. We will later we got a second product, and that company was not so deciding on very high number of bottles. They only said 500, so that was good. But this company minimum must be 1,000 bottles. Okay, so we did a lot of efforts to start selling the 1,000 bottles. We created a web page for the company. We created company FB. I think this one is the company FB. You can see uh, got already 556 followers by then after advertising aggressively. Uh, However, a lesson learned, we started off by doing unpaid online marketing with all these online tools. And what we found out was doing unpaid was a waste of time, and also does not have the view or the effectiveness that we wanted. So, either way, I just show you some of the unpaid advertisement that we've done. This one with eBay, unpaid, no visibility, no purchases as well. Okay. This is our company profile, the front page of our website, done very professionally, no problem. Okay. Having learned the lesson that unpaid is not going to create the visibility, we went all paid online marketing, and also we created this brochure. The brochure was helpful to put into pharmacies, small pharmacies, to go and when exhibition comes, we use the brochure to give to the customer, potential customer, and then later they read the brochure in Bahasa and also in English, and then they will call up for more clarification. Same thing with paid online marketing. Doesn't matter Shopee, FB ad, or Muda. All after we created the advertisement, definitely there will be lots of question, and then only finally sales will take place or closing. The advertisement for paid look like in Shopee, and as you can see, you've got 100 pages for what item is this? If you put it as men, search result for men, 100 pages, but after you pay, it's already in the first page itself, the product, our Tongkat Ali product. Here, just to give you an idea, on Muda also we paid, and then the price was put at 59. You'll see another product at 79. Always have the 9. 9 is meant for the post laju package that we have to buy in order to send by post laju. Okay, so the actual price for this product is 50. So after some time, we were starting to get sales and then returning customers. There's even a customer that's been with us buying this product for past 2 to 3 years already. Okay, so this is to give you an idea how much we spend. There were two grants. One is called commercialization grant within the government university funded, and there also an external grant called MTUN grant given by Malaysian Technology University Network, comprising of 4 universities that actually fund this external grant. So we had that grant as well, and we use all that to sell the products. There were two to also register. So all the money done for registration, to start the processing of the raw material, to buy the raw material, and also to send it to contract manufacturers, pay them, everything included in this 80,000. And finally, two products were ready with ministry approval. The first one is called Nina Square Tongkat Ali capsule, 260 milligram. Um, we price it at 69, but sometimes we have promotion and sell it at 59 only. While the second product, a bit more different, where it is a mixture of three Tongkat Alis, all the three, white, black, and red combined together, and we call it Bio Root to Ali capsule, and that is priced at 79 ringgit per bottle of 60 capsules. So in conclusion, this is the technology readiness level, the original one from NASA at 1970. This is how it looks like. They probably, after landing in the moon in 1969, decided to be more effective, more systematic. They came up with this until got launch, system test, launch, and operation. But I mostly has a TRL level, but for my project, I customized or modified it to be for my project of Tongkat Ali. So, firstly, I identified problem statement, as in there is a need because there's many fake products out there. We tested 50 randomly selected products, and then we found out four of it is fake. So we need to be more researchers need to come up with authenticated tongkat ali and make it into capsule. Therefore, customers have certain trust to the researchers. So that's what we did, problem statement. And then we also realized uh, tongkat ali has been wrongly marketed as a sexual performance, sexual dysfunction, libido product rather than for the purpose of increasing testosterone. So we changed the understanding market strategy to actually say it is to elevate testosterone for aging men. Therefore, wider customer range rather than focusing on sexual performance, sexually improving. That one smaller uh, market. And then TRL 3, 4, and we demonstrate the product, the prototype, whether it can sell or not, whether it can be done or not in the laboratory, whether it can scale up or not. Those are the one, this TRL. And then we went to TRL 8 to register and get approval from ministry. Therefore, we don't have to worry putting it in sale everywhere. So it won't be confiscated because it's an illegal product. And finally, TRL 9, we launched it officially. So if you ask me which one is most tedious and lengthy, that will be TRL 8 because surprisingly, I didn't expect it to take that long, 6 months to 1 year. I thought in 1 month can get approval, but there's a lot of queue from other products also trying to get approval, I assume. Okay. And then which one challenging? TRL 9, because once you come up with a new product, you always have that anxiety whether people will buy or not, especially when you are working on a plant that nobody heard of. But I'm lucky, I am actually using Tongkat Ali, and it's not only popular in Malaysia, not only popular in surrounding countries, but world over. So easy to sell because of that. And also got returning customer. And after doing everything from A to Z, from production, sourcing out the raw material, and then even doing sales, I decided to give up on one area of the whole A to Z, which is marketing. I think the university should have their own unit of salespeople trained, capable in selling this product, and therefore I suggested as such. And this whole presentation, this the whole process from A to Z for both of the products, the Bioruto Ali capsule and also the first product, which is only having Tongkat Ali Putih or Uricoma longifolia. So we started from getting the source, reliable source for the raw material, processing the raw material, up to testing it for quality assurance, finally putting in the bottle, the capsule. But we are not stopping there. Hopefully, in the future, we already found a way to remove all other phytochemical and leaving behind only the active constituent related to boosting testosterone. That will be good because it's going towards the direction of drug development, where we will now no need to have a very big capsule. It will be sufficient to make it into tablet, and then later after much safety and efficacy studies on that particular constituent, on the lead compound, it's possible to have drug category.